Hello and welcome to BioStock Studio. Today we're putting the spotlight on cancer vaccines. And joining me to help us learn more is Sara Mangsbo, Chief Innovation Officer at Ultimovacs. Sara is also Research Director and Associate Professor of Immuno-Oncology at Uppsala University. Thank you for joining us today for this conversation, Sara.
Thanks for having me.
Well, I'd like to begin to talk a little bit about your professional background, specifically your experience within cancer immunotherapies.
Yeah. So, I've been in this field for a long time. I started working within cancer immunotherapy since 2006, I think. And at that time, I think we sort of were kind of a few enthusiastic persons in the field. Mm-hmm. Not many going to conferences, very few actually coming to your poster at the presentation event. And then, boom, cancer immunotherapy grew. And I think that all, of course, came with the known checkpoint inhibitors, the ones that sort of boost our T-cell immunity and helps our immune system to target cancer. And that transformation, seeing that and being part of that transformation where not only academic parties sort of talked about cancer immunotherapy, but this was something that came as an interest for the industry as well. That was amazing to see. And I think at least how I experienced that was the fact that you just didn't help people to tackle the tumor for a short while, but you actually made a difference in survival for patients. Mm-hmm. And I think that transformation went through the whole society. Of course, patients experienced that, but also caregivers and the ones that treated the patients, and yeah, it was amazing to see.
Mm-hmm. Going deeper into what a vaccine is, the pandemic, which is unfortunately still ongoing, has brought a lot of questions and confusion about what a vaccine is exactly. And considering your experience, could you tell us and answer the question today, what is a vaccine?
Yeah, so what I talked about before, checkpoint inhibitors, they are sort of allowing us to keep and maintain a functional immune system against the tumor. But then the tumor has acted as a vaccine, because a vaccine is actually as a substance that instructs the immune system and teaches the immune system to develop a memory response. So we call this antibodies or T-cells that can target a disease. And what we want to achieve with a vaccine is either to prevent a disease or to actually treat a disease. And vaccines can be used either off, but we have to design them a bit differently depending on if we want to prevent a virus to come into a cell and prevent the disease to occur at all, or if we want to treat a disease that is already there, then something is wrong within the cell and the tumor cells are growing, and then we have to tackle it with T-cells.
Mm-hmm. You mentioned vaccines against viruses. Vaccines against pathogens in general have existed for hundreds of years and have been very effective. Why is it taking so long to develop an effective vaccine against cancer?
Because those substances that we use to teach the immune system, it's a lot easier to teach the immune system to recognize something that comes as a foreign pathogen. And when we are trying to reteach almost the immune system to recognize something that was actually our own not harmful tissue from start, then there's a problem, because we already educated the immune system to not target that tissue. Yeah. And then we have to sort of redo that teaching, and that can be a bit difficult. If the tumor is very different from normal tissue, then it can be a bit easier, and then we can tackle the tumor with these checkpoint blockers that just ensures that those T-cells that are taught up by the tumor just functions better. Mm-hmm. But when we can't do that, maybe then we go into other immunotherapies like vaccines, where we try to teach new T-cells that we can maintain then with checkpoint inhibitors.
Okay. You are Chief Innovation Officer at Ultimovacs, which is a Norwegian biotech company that is developing its own cancer vaccine technology. I was hoping you could tell us a little bit more about this technology.
Yeah, so I think Ultimovacs has something that can be used and harnessed for many different cancer forms. So what we are trying to do is then to teach our T cells to recognize the tumor. And the hard part was, what do we want to teach with, right? So Ultimovacs has a set of small substances, we call them peptides, puzzle pieces almost. And the tumor needs something to grow. It needs something to grow indefinitely. And that is that it can't really outgrow its own code. So when it divides and divides and divides, then the code that instructs the cells what proteins to maintain and so forth, that code tends to get shorter on the ends of the code, the DNA. And then we have to upregulate something called telomerase in the cells so that we maintain those ends. And the tumor is excellent at doing that. That's the way the tumor can maintain the growth. But then we thought, why not harness that, right? So why not try to use that activity that the tumor has and then say that this is what we can teach the immune system to recognize, because that's what the tumor needs, a little bit of that protein, that telomerase. And then we take small parts of that telomerase. And those parts cannot induce any disease. They cannot function in other cells to do that. They can just teach the immune system to recognize those cells that have an unnatural activity of that protein.
And these parts you're talking about, are they expressed in all cancers or just a subset of cancers?
So it can vary a little bit, but usually one talks about 80 to 90% of tumors have an activity of telomerase. But then there are some tumors which have less and some tumors that have more. But it seems to be a possible universal vaccine, although we have to recognize that every tumor is unique.
And Ultimovacs also has another platform, an adjuvant technology platform. Could you tell us a little bit more about that one?
Yeah, so this is something that I have sort of brought from start together with colleagues in the Netherlands when I was in academia there. And we sort of started off with, how can we improve these small puzzle pieces? How can we improve their function, their sustainability, and their ability to induce an immune response in the body? And so we thought one way of improving that is to attach these tumor pieces to something that is foreign, that is from another protein, which is a toxin protein actually. And then when we do this, then the immune system already has a previous experience of this. So then the immune system reacts instantly and tags along the tumor parts. So we're just trying to boost the vaccine a little bit with this technology actually, and thereby improving the number of T cells we can make with the vaccine.
Very interesting. So in that sense, it would become a little bit more similar to what a vaccine against pathogenic disease might look like, let's say, because it comes, the source comes from outside.
Yeah, a little bit. And then we tag in the other part. But we don't want to use too much of the sources outside, because then the risk is that we push the immune response towards that. So this is just a small part of it.
Fair enough. Well, finally, companies like Moderna, which became well known for having developed one of the mRNA-based COVID vaccines, they're also developing the same technology for cancer vaccines as well. First of all, what are the pros and cons of this? And secondly, how would this compare to what Ultimovacs is doing?
So everything is about this puzzle piece that we're going to instruct the body to react to. And that's always something which is usually a peptide or a protein. So a protein is based out of peptides or amino acids. But you can either make those peptides in, the lab, or you can ask the body to make them. So what we have seen is a transformation from making those peptides in the lab towards allowing cells to make them. what they had to do to achieve that was also to make them less immunogenic, less prone for the immune system to react to the RNA itself. But otherwise, the principle is actually similar. What we are doing, we're using peptides, they are using the code, the mRNA, to make the peptides. It's not very different. And then everything needs to be hooked up with something that also stimulates the immune system at the same time to lead to this sort of danger signal, like now we're gonna induce an immune response.
Okay.
So I would say they're quite similar, just a bit of a difference in how we make the drug.
All right, very interesting. Well, thank you so much for answering the questions and for having this conversation with me today. And yeah, let's hope your technology can become a treatment very soon.
Thank you.